Just confirm me once whether my voice is audible or not. Please confirm it once. Just confirm it once whether my voice is audible or not. Am I audible to you? Okay. So a very good evening everyone. Myself Prateek Mishra and I welcome you to the YouTube platform of IMS GATE Academy. We are conducting this series of most expected question. Thank you so much Nikhil. We are conducting this series of most expected question in GATE exam. We are discussing about well hydraulics, sedimentation analysis, alkalinity and hardness, disinfection. Important topics we have seen that are expected in environmental engineering in GATE-24. Today we will going to see one of the very important topic, and that is biochemical oxygen demand, BOD. A very good evening Nikhil, very good evening. Today we will going to see a very important topic of biochemical oxygen demand. Which is BOD. Okay. Now you have to understand this. When we talk about wastewater engineering, once you have supplied water to the community after different usages, this water is converted into wastewater. The collection of this wastewater, transportation of this wastewater up to sewage treatment plant, then treating this wastewater, and after treatment disposing this treated sewage is termed as wastewater engineering. Again, we need to give certain degree of treatment to this wastewater. Then how to decide how much treatment to be given to this wastewater? So that treatment is decided on two factors. First one is quality of wastewater or quality of sewage, and second one is the self-purification capacity of the source, where we will going to dump this treated wastewater, right? In order to check the quality of wastewater, the parameters that are used are wastewater quality parameters. A very good evening Tushar, very good evening. Okay, so the parameters which are used to analyze the quality of wastewater are termed as wastewater quality parameters. Wastewater quality parameters are again of three types: physical wastewater quality parameters, chemical wastewater quality parameters and biological wastewater quality parameters. All the water quality parameters that we have studied for raw water engineering are common in wastewater engineering. Apart from that, there are certain more wastewater quality parameters that we have to see, especially for wastewater engineering. In wastewater engineering, the additional wastewater quality parameters that we need to study is chemical oxygen demand, COD. Then you have biochemical oxygen demand, BOD. Then you have theoretical oxygen demand, THOD. Then you have total organic carbon, TOC. Then you have population equivalent. And lastly, you have relative stability. So these are the additional wastewater quality parameters that we have to learn. Right, sir, physical are same in both. Yes, Tushar, physical are same in both. Suspended solids, turbidity, taste and odor, color, temperature, all those parameters are same for wastewater and for raw water engineering. In chemical water quality parameters, whatever chemical water quality parameters that we have read for raw water engineering are common with wastewater engineering. In wastewater engineering, these are the additional chemical water quality parameters that we have to see. COD, chemical oxygen demand, BOD, biochemical oxygen demand, then THOD, theoretical oxygen demand, TOC, total organic carbon, population equivalent and relative stability. Out of all these chemical water quality parameters, your biochemical oxygen demand, that is BOD, is very, very important, right? And today we will going to see what do we mean by biochemical oxygen demand, how we calculate biochemical oxygen demand, and today I am going to rectify all your conceptual doubts regarding this. Okay, so let's start with understanding with what do we mean by biochemical oxygen demand. My dear students, one thing I would like to tell you, when I talk about sewage, this sewage contains organic matter as the major impurity. Okay, in sewage, the major impurity that is present is organic matter. Now, this organic matter can be of two types. This organic matter which is present in sewage can be of two types. Organic matter can be of two types. The first type of organic matter is biodegradable organic matter. Biodegradable organic matter. This is that type of organic matter which, if it comes in contact with any microorganisms, microorganisms are capable to degrade this biodegradable organic matter. These are simple organic matters. Okay. The next type of organic matter is known as non-biodegradable organic matter. The next type of organic matter is non-biodegradable organic matter. The next type of organic matter is non-biodegradable organic matter, right? In order to decompose this non-biodegradable organic matter, you need to add chemicals, especially the oxidizing agent, to remove these non-biodegradable organic matter from the sewage. Okay. So, the organic matter present in the sewage is of two types, biodegradable organic matter and the non-biodegradable organic matter. All these types of organic matter will be removed by oxidation or by microorganisms. In order to oxidize this biodegradable organic matter, you require some amount of oxygen. In order to oxidize this non-biodegradable organic matter, again you require some amount of oxygen. Now here I will define biochemical oxygen demand and COD. The amount of oxygen required to oxidize only biodegradable organic matter is termed as biochemical oxygen demand. The amount of oxygen required to oxidize only biodegradable organic matter is termed as biochemical oxygen demand. It is termed as biochemical oxygen demand. It is termed as biochemical oxygen demand or BOD. Simple. The amount of oxygen required to oxidize only biodegradable organic matter is termed as biochemical oxygen demand. Now next, the total amount of oxygen required to oxidize biodegradable plus non-biodegradable organic matter is termed as chemical oxygen demand COD. The total amount of oxygen required to oxidize biodegradable plus non-biodegradable organic matter is termed as COD. It is termed as COD or chemical oxygen demand or chemical oxygen demand or chemical oxygen demand. Simple. So basically both of them are oxygen demand. The amount of oxygen required to oxidize only biodegradable organic matter is termed as biochemical oxygen demand, and the total amount of oxygen required to oxidize biodegradable plus non-biodegradable organic matter is termed as chemical oxygen demand. By this definition, can you just quickly tell me which one will be more? COD will be more or BOD will be more? Please comment quickly which chemical oxygen demand will be more. COD will be more or BOD will be more? Comment and tell me which demand will be more. COD will be more or BOD will be more? Quickly comment your answers. Let me see how many of you judged this correctly. Which demand will be more? It is totally the amount of oxygen required. The amount of oxygen required to oxidize biodegradable organic matter is BOD. And the total amount of oxygen required to oxidize biodegradable plus non-biodegradable organic matter is COD. So by obvious reasons, COD is always more than BOD, right? COD is always more than BOD. COD is more than BOD. COD is always more than BOD. Now, if in the system or if in the water sewage sample only biodegradable organic matter is present, then that is the only case where COD will be equal to BOD. When only biodegradable organic matter is present, then only in that case your COD will be equal to BOD. In all the other cases, COD will always be more than BOD. So there is one relation which says that COD is greater than equal to BOD. COD is greater than or equal to BOD. And I think you have got the proper reason for this. COD is the total amount of oxygen required to oxidize any type of organic matter, and BOD is the amount of oxygen required to oxidize only biodegradable organic matter. Okay. Now coming to this, test for BOD. Test for BOD. Test for BOD. How do we determine the BOD of the water sample, right? So first what you need to do is, you need to take some volume of wastewater sample. You need to take some volume of wastewater sample. The some volume of wastewater sample, say X ml of wastewater sample. You took X ml of wastewater sample. And this X ml of wastewater sample is mixed with some amount of aerated water. In this X ml of wastewater sample, we mix it to form a diluted sample. In this X ml of wastewater sample, some Y ml of aerated water is added. Some Y ml of aerated water is added to have the total volume as X plus Y ml. This is the total volume of X plus Y ml of the diluted sample. So this is my diluted sample. This is my diluted sample. Now usually the student has this confusion, why we are adding diluted sample. Okay, first of all, what we want to analyze? We want to find out what is the BOD of the water sample. That is, how much amount of oxygen is required to oxidize the biodegradable organic matter present in the water sample. For that, this aerated water is added in order to provide sufficient oxygen so that degradation of biodegradable organic matter can happen, or decomposition of biodegradable organic matter can happen. So you have X ml of wastewater sample, it is mixed with Y ml of aerated water to form X plus Y ml of diluted sample, right? To form X plus Y ml of diluted sample. Now this diluted sample, for this diluted sample, you need to do two things. First, you need to measure the dissolved oxygen concentration, which is known as DO initial. It is initial dissolved oxygen. It is initial dissolved oxygen. Initial dissolved oxygen. It is initial dissolved oxygen of this diluted sample. Okay, the first thing that you need to do is, you need to measure initial dissolved oxygen of this diluted sample. The second thing you need to calculate is the dilution ratio for the dilution that you did. So dilution ratio dr is equal to dilution ratio dr. I'll write here dilution ratio dr is equal to final volume of the diluted sample, which is X plus Y ml. The final volume of the diluted sample is X plus Y ml, divided by the volume of original sample present in the diluted sample. So in this x plus y ml of total volume, x ml of original wastewater sample is present. So dilution ratio is total volume of the diluted sample divided by the volume of original sample present in the diluted sample. So this is x plus y divided by x. These two things you need to calculate. Again, we will start the test. How you should start the test? Take x ml of wastewater. In that x ml of wastewater, add y ml of the aerated water, so that sufficient oxygen you are supplying for the carrying out the decomposition of biodegradable organic matter. Once this diluted sample is formed, volume is x plus y ml. For this diluted sample, measure the dissolved oxygen, which is termed as the initial dissolved oxygen of the diluted sample. The second is mark the dilution ratio. Dilution ratio is the ratio of final volume of the diluted sample divided by the volume of original sample present in the diluted sample. That is the dilution ratio, right? Now, once this is done, this diluted sample, it is placed in the BOD bottle. This diluted sample is placed in the BOD bottles. So, place diluted sample in the BOD bottle. Place diluted sample in BOD bottles. Place diluted sample in BOD bottles, and these BOD bottles have a volume of 300 ml. They are standardized, right? Directly in lab, if you ask someone where are the BOD bottles, so they can clearly give you this BOD bottles. Volume is fixed, 300 ml. So this diluted sample is poured in the BOD bottle and the lid is closed. So this BOD bottles looks like this. This BOD bottles looks like this. You just close the lid, right? This kind of lid is there for the BOD bottle. This is the glass lid, right? You just close the lid. The total volume of the sample is 300 ml. And inside this BOD bottle, you have placed the diluted sample. You place the diluted sample, right? Now these BOD bottles are placed inside the incubator They are placed inside the incubator. They are placed inside the incubator at 20 degrees Celsius for 5 days. And we should ensure that the incubator are placed in dark place. They are placed in dark place, right? Reason I'll tell you in short while. Right now, you have to just focus on what we are doing for performing the test. You place the diluted sample in the BOD bottle, which is having the volume of 300 ml. Now, this BOD bottle with diluted sample is placed inside the incubator at 20 degrees Celsius for 5 days in the dark place. At this temperature of 20 degrees Celsius and for this 5 days, the oxygen that we have added from our side through aerated water, this oxygen will carry out the decomposition of biodegradable organic matter which is present in the diluted sample. And the value of dissolved oxygen, which was DO initial early, the value of dissolved oxygen will keep on falling as the number of days will pass, because this dissolved oxygen is getting used up for carrying out the decomposition of the biodegradable organic matter. Okay, again I'll repeat myself. Place this BOD bottle in an incubator at 20 degrees Celsius for 5 days. So the dissolved oxygen which is there in the sewage will carry out the oxidation of biodegradable organic matter and will get used up. After 5 days, what you have to do is, after 5 days, take out this diluted sample. After 5 days, after 5 days, you take out this sample and measure the final dissolved oxygen value. Measure DO final and measure DO final, that is final dissolved oxygen value. Final dissolved oxygen value, right? So I think the test is very simple. The first thing is prepare the diluted sample. For that diluted sample, measure the initial dissolved oxygen and measure the dilution ratio. After that, place that diluted sample in the BOD bottle, which is having the volume of 300 ml, and place this BOD bottle inside incubator at 20 degrees Celsius for 5 days. Within these 5 days, the dissolved oxygen that is present in this sewage sample or diluted sewage sample, this dissolved oxygen will get used up for carrying out the decomposition of biodegradable organic matter. For 5 days, this oxygen will be getting used up for this decomposition. After 5 days, you measure the final DO level, right? Now this difference in initial DO and final DO, the difference in initial dissolved oxygen and the final dissolved oxygen will going to tell us how much amount of dissolved oxygen is used for carrying out the decomposition of biodegradable organic matter. How physically we measure DO final? Beta, DO final is measured using Wrinkler's test. Remember, Nikhil, we have discussed about Wrinkler's test, right? So Wrinkler's test is performed in this, the sample that we have taken out after 5 days. It's a very good question, how we physically measure DO final. So DO final or DO initial, both the DOs are measured using Wrinkler's test. Okay, Wrinkler's test is again a topic of discussion for next 20-30 minutes. So Wrinkler's test is directly, I can say the name, it is used to measure the DO final value and DO initial value. Anytime if we need to measure dissolved oxygen level in the water, we go with the Wrinkler's test. Okay. Sir, again we need to conduct that test. Okay, yes, we need to conduct that test every time. Whenever we want to measure the dissolved oxygen level, we go for the Wrinkler's test. Every time we perform the Wrinkler's test for measuring the dissolved oxygen level. Okay. So DO final is measured. The difference in DO initial and DO final will tell us about how much amount of oxygen is used to decompose the biodegradable organic matter. The final BOD of the water sample is reported like this. So BOD of wastewater sample, BOD 5. 5 is the number of days of incubation and the temperature of incubation is 20 degrees Celsius. So this is the standard way of representing the biochemical oxygen demand. BOD 5 means 5 days of incubation at 20 degrees Celsius. This is equal to DO initial. It is equal to DO initial minus DO final multiplied by the dilution ratio. Multiplied by the dilution ratio. DO initial minus DO final multiplied by the dilution ratio will be BOD 5 at 20 degrees Celsius. Okay, now you have to understand this very carefully. There are 2-3 major aspects for this expression. First thing, I'll start with this. Why it is kept in the dark place? Okay. For example, if algae is present in your sewage sample and if this incubation or incubator is kept in a place where sunlight is coming, under the action of sunlight, this algae will start undergoing photosynthesis. And during these 5 days of incubation, this algae will keep on releasing oxygen. Okay. In that case, this final DO that we will report, it will be more than actual. And if final DO is more than actual, the difference in DO initial minus DO final is less than actual. So if we place this incubator in a place where sunlight is coming, then the algae present in the sewage sample will undergo photosynthesis that will release oxygen, and in that case the reported DO is more than actual, and the BOD that we calculate will be less than actual. And if we calculate BOD less than actual, then it will affect the subsequent treatment process, because all the wastewater treatment process is designed on this basis that how much amount of organic matter is present. If we are reporting less value of BOD more than actual, then actually we are reporting less value of organic matter present in the wastewater. And if we report less value of organic matter in the wastewater, then actually we are not designing the treatment process efficiently. Some part of the untreated organic matter will come out, and that will cause very unsanitary situation wherever this treated sewage is disposed. So every time when we go with calculation of BOD, we place this incubator in dark place so that this algae which is present inside the sewage may not come in contact with the sunlight. And if it doesn't come in contact with the sunlight, it will not undergo the process of photosynthesis. Okay. So this is how we measure BOD5 at 20 degrees Celsius is equal to DO initial minus DO final multiplied by dilution ratio. Okay. Now the second thing. BOD is amount of oxygen required to oxidize the biodegradable organic matter. Means we are relating oxygen with the biodegradable organic matter. Oxygen is related with biodegradable organic matter. Biodegradable organic matter. It means that if I am reporting BOD, I am indirectly reporting the organic matter. BOD is just an indirect way of reporting the organic matter. If I am reporting BOD, it means I am reporting the organic matter. More the BOD of the water sample, more will be the organic matter present in the sewage. As simple as that. So every time when you hear about BOD, just think about it is related with the organic matter. More BOD, more organic matter. Less BOD, less organic matter. So instead of telling organic matter directly, we represent it through BOD. And BOD is defined as the amount of oxygen required to oxidize the biodegradable organic matter, right? So please keep a check on this. Oxygen is related with biodegradable organic matter. BOD is related with the organic matter. More the BOD, more will be the organic matter. Okay, after this, next is kinetics of organic matter. Kinetics of organic matter. So basically we want to discuss how the biodegradable organic matter will going to decompose under the action of microorganisms. So next topic of discussion is kinetics of organic matter. On the x-axis, I have plotted the oxygen equivalent of the organic matter, and on the y-axis it is time. On the x-axis it is oxygen equivalent of organic matter. Denoted by N, right? N is the oxygen equivalent of the organic matter. Example is, for example, 10 milligram of organic matter is present. So either I can represent this organic matter in the form of its weight, or I can represent the same organic matter through the amount of oxygen required to oxidize this organic matter. And if we represent this organic matter through the amount of oxygen oxidize this organic matter, it is known as the oxygen equivalent of the organic matter. Initially, the amount of organic matter present is N0. Initially, the amount of organic matter present is N0, or I'll represent this with L, not N. I'll represent this with L. I'll represent this with L. initially the amount of organic matter which is present is L naught, okay? And over the period of time, under the action of microorganisms, this organic matter keeps on reducing like this. This organic matter keeps on reducing like this. This organic matter keeps on reducing like this, right? L naught is the total amount of organic matter initially present. And this organic matter keeps on reducing like this. After any time t, the amount of organic matter that is present in the system is LT. After any time t, the amount of organic matter present in the system is LT, okay? So at t is equals to 0, the amount of organic matter present in the system is L naught. And at t is equals to t, the amount of organic matter present in the system is LT. And clearly from this curve, we can see that the organic matter is reducing in the system. Now this reduction or the decomposition of this organic matter follows first order kinetics. So you can write down decomposition of organic matter follows first order kinetics. Decomposition of the organic matter follows first order kinetics. And if I have to write the decomposition of organic matter, it is the rate of change of organic matter with respect to time, dLt by dt. And why minus sign is placed? Because this organic matter is reducing with time. So minus times of dLt by dt, this is directly proportional to the organic matter present at any time t. Means the rate of change of organic matter or the rate of change of or the rate of decomposition of the organic matter will depend upon how much amount of organic matter is present. Clearly from this curve you can see that initially if more organic matter is present, the rate of decomposition is more, and as the organic matter is decreasing, the rate of decomposition or the slope of this curve is constantly reducing, right? Here the slope is more, and then subsequently the slope is reducing, and that we what mean by rate of decomposition. So it is directly proportional to the amount of organic matter present after any time t. So just remove the proportionality sign. You have dLt by dt is equal to k into LT. K into LT. Just rearrange this. So minus dLt by LT is equal to k into dt. Integrate this expression, take minus sign that side. So you have dLt by dt is equals to minus k into dt. Now integrate this. At t is equals to 0, the amount of organic matter present is L naught. At t is equals to t, the amount of organic matter present is LT. When you integrate this expression, you will get the expression of amount of organic matter that is present in the system after any time t. So you will get natural log of LT by L naught is equal to minus k into t. Okay, take the anti-log. So you have LT is equal to L naught e raised power minus k into t. This is one of the very prominent expression that we use, or from where the questions can be framed. LT is equals to L naught is equals to e raised power minus k into t. And what is LT? LT is the amount of organic matter which is remaining in the system after any time t. So you can clearly observe from this curve, this is your LT, and this LT is nothing but your organic matter remaining, which is known as BOD remaining. This is your BOD remaining. So LT is nothing but the organic matter remaining in the system after any time t, which is also known as BOD remaining. So from here we can write down BOD remaining is equal to L naught into e raised to power minus k into t. Now further I want to tell you about this L naught. What is L naught? L naught is the total amount of organic matter present in the system. This is L naught, the total amount of organic matter present in the system. And this L naught is known as the ultimate BOD. L naught is known as the ultimate BOD. That is the total amount of organic matter that is present in the system. So L naught is ultimate BOD. And what is ultimate BOD? Ultimate BOD is the total amount of organic matter present in the system. Ultimate BOD is total amount of organic matter present in the system or present in wastewater. L naught or BOD ultimate is same, so it is denoted by BODU. So what is BODU? BODU is ultimate BOD, which tells us about the total amount of organic matter present in the system. Now can you tell me, it doesn't matter whatever decomposition is happening in the sewage, will it change ultimate BOD? Can ultimate BOD be changed with decomposition? Ultimate BOD is simply the measure of total amount of organic matter present in the system. So ultimate BOD of the water sample is fixed, it is constant, it will not change, right? BOD ultimate tells us about the total quantity of organic matter which is present in the system. Now we can just rewrite the BOD remaining expression. So BOD remaining after any time t is equal to BOD ultimate into e raised to power minus k into t, right? BOD remaining is equal to BOD ultimate into e raised to power minus k into t. Now observe this very carefully. So natural log of LT by L naught is equal to minus k into t. If I need to convert this natural log to the base 10, so this is 2.303 log to the base 10 of LT by L naught is equal to minus k into t, right? Take this 2.303 this side, so you have log to the base 10 of LT by L naught is equal to minus k by 2.303 into t. Now this constant k by 2.303, we name it as a new constant which is kd. Okay, and when you take the NT log, you will get one more expression for BOD remaining. So you have LT is equals to L naught into 10 raised to power minus kd into t. Okay, so again you need to keep a check on this. Whenever you are using k, you have to use base e, and whenever you are using kd, you have to use base 10. it will be clearly mentioned in the question whether the value of K is given to you or KD is given to you. Okay. Now from this expression we can write down BOD remaining after any time t is equal to BOD ultimate into 10 raised power minus KD into T. BOD ultimate into 10 raised power minus KD into T. So this is the first expression which tells us about BOD remaining. That is the amount of organic matter which is remaining in the system after time T. Okay. Now next thing that I would like to ask is how much amount of organic matter is consumed or decomposed. So the amount of organic matter which is decomposed or consumed, it is this value. This is the amount of organic matter. This is the amount of organic matter which is decomposed. And this is known as BOD consumed. This is known as BOD consumed after time T. This is known as BOD consumed after time T, which is the difference of ultimate BOD minus BOD remaining after time T. BOD consumed after time T is the difference of the organic matter which was initially present minus the organic matter which is left in the system after time T. So BOD consumed after time T is equal to BOD consumed after time T is equal to L0 minus LT. Okay. Now this L0 minus LT can be written as L0 minus LT can be written as L0 into e raised power minus K into T. Okay. L0 minus LT can be written as L0 minus L0 into e raised power minus K into T. This L0 if you take out common, you will have BOD consumed after time T is equal to L0 common into 1 minus e raised power minus K into T. Okay. Now this L0 is nothing but BOD ultimate, BOD consumed after time T is equal to BOD ultimate into 1 minus e raised power minus K into T. So this is the expression of BOD consumed. BOD consumed is BOD consumed after any time T is equal to BOD ultimate is equal to BOD ultimate into e raised power minus K into T. Again, if you use to the base 10, you have to use KD. So BOD consumed after time T is equal to BOD ultimate into 1 minus 10 raised power minus KD into T. 1 minus 10 raised power minus KD into T. Now this K and KD are known as deoxygenation rate constant. K is for the base E and KD is for the base 10. K and KD are deoxygenation rate constant, which tells us about the rate of decomposition or degradation of the organic matter. If the rate of degradation of the organic matter is more, it means the organic matter is a simple chain organic matter. The decomposition of the organic matter is happening very easily. That is what we understand through deoxygenation rate constant. If the value of K for a particular organic matter is more, means the nature of organic matter is more complex. It will take more time for getting degraded. If the value of K is less, it means it is a simple organic matter and it can be easily degraded. Okay, so K and Kd are the deoxygenation rate constant. Then we have expression for BOD remaining. BOD remaining after time t is equal to BOD ultimate into 1 minus e raised power minus k into t, and the same is equal to BOD ultimate into 1 minus 10 raised power minus kd into t. BOD ultimate is equal, BOD remaining is equal to BOD ultimate into 1 minus e raised to power minus k into t, or it is also equal to BOD ultimate into 1 minus 10 raised to power minus, sorry, this is BOD remaining, very sorry. BOD remaining after any time t is equal to BOD ultimate into e raised to power minus k into t, or it is equal to BOD ultimate into 10 raised to power minus kd into t. This is the first expression. The next is BOD consumed. BOD consumed after any time t is equal to BOD ultimate into 1 minus e raised to power minus k into t, where t is the time in days. Always remember, the value of deoxygenation rate constant will be always in per day. So BOD consumed after any time t is equal to BOD ultimate into 1 minus e raised to power minus k into t, or it is also equal to BOD ultimate into 1 minus 10 raised to power minus kd into t. 1 minus 10 raised to power minus kd into t, right? These expressions should be known to you, and these are very easy expressions, right? Now I'll give you a very short trick, very short trick, and that trick is which BOD we need to calculate in the exam. If it is asked to calculate BOD of the water sample, okay, so there will be three things that are mentioned. First will be ultimate BOD. If you are asked to calculate the ultimate BOD, then this is the value. This is the ultimate BOD that you need to calculate, BODU. Second, if nothing is mentioned in the question and they are asking you to calculate BOD of the water sample after 5 days, after 3 days, after 8 days, right? What I am saying, if they are only asking calculate BOD of the water sample after 5 days, it means that you need to calculate this BOD consumed every time. If the question is saying that you need to calculate BOD after 5 days, it clearly means that you need to calculate this consumed BOD. Last but not the least, if they are expecting you to calculate BOD remaining, they will clearly mention calculate BOD remaining after 5 days. In that case, you need to measure this one. So if nothing is mentioned, only this thing is mentioned, that calculate BOD after 2 days or 5 days, you always have to calculate BOD consumed. For ultimate BOD it will be mentioned, and for BOD remaining again it will be mentioned. But if nothing is mentioned, it is mentioned like calculate BOD of water sample, calculate BOD of water sample after 5 days, right? If this kind of thing is given to you, always you need to calculate BOD consumed, always, right? If nothing is mentioned in the question, you have to calculate BOD consumed. For ultimate BOD and for BOD remaining, they will clearly mention in the exam. Now the next confusion comes whether to use this expression, whether to use this expression. So whenever you are calculating BOD consumed or normal BOD, you will always use this expression. For BOD remaining, this is the expression. The third confusion that comes is whether to take e raised to the power or whether to take 10 raised to the power, right? So if the value of k is given to you, it will be again clearly mentioned in the question which base you need to use. Or if it is not mentioned and only k is given, then you have to use base e. If kd is given, you have to use base 10. As simple as that. So these are the common areas from where there can be doubts regarding your BOD, right? So this is the total topic of BOD that we have discussed. Just by moving ahead, right, the last thing I want to ask you in simple words. Remember, we have calculated BOD using this test. We have calculated BOD using this test. BOD 5 is equals to DO initial minus DO final multiplied by dilution ratio. This is which BOD? BOD remaining or BOD consumed? I'll wait for your answers. This is which BOD? BOD remaining or BOD consumed? So basically, we are taking the difference of the dissolved oxygen. Means how much amount of dissolved oxygen is getting utilized for carrying out the decomposition of the organic matter. So in this test, basically we are measuring BOD consumed. So the test of BOD will always give us BOD consumed. Okay. The test of BOD will always give us BOD consumed. So by these words, I would like to just end this session. I hope this session was highly informative for you. It is a very important topic and n number of questions are asked in GATE and other technical exams. So this is all what I have to deliver for now. Please join our Telegram group. The link is in the description box for daily quizzes and for getting the updates of all the YouTube sessions that we are having. We are running these absolutely fantastic sessions of most expected question and the excellent practice questions for GATE-24. So I wish all the aspirants to see all our video lectures because they are highly precisely designed and they are designed to give you the best of the content in limited time. So this is all of what I have to say. Myself, Prateek Mishra, and I'll signing off for today. And I'll see you in the next session with a very, very important topic in the next session. Thank you so much for listening patiently. Have a very good night ahead. Thank you so much.